Hello students, so once again I welcome you on our YouTube channel Little Kingdom School. As we discuss the chapter 1 sexual reproduction in flowering plants in previous chapter, so today we are going to continue with that topic. Done. So lastly we discussed about the anther and what is microsporangia. So let's quickly revise the previous topic. So first of all start with the flower. As we know now, flower contain 4 basic part: androecium, which is a male reproductive part; gynoecium, female reproductive part; petal and sepal. Clear? So today we are going to discuss further about androecium. So as we know, androecium contain 2 parts. First, filament and anther. Filament is a long, slender-like structure which provides support to the anther, and anther is a bilobe structure like this. Quickly revise. So anther is look like this. This one is filament, this one is anther. Anther is a bilobe structure. If we take a thin section of the anther, it will look like this. We already discussed in previous lecture. It will look like this. Done. The single anther contain 2 lobes, first lobe, second lobe. Each lobe contain 2 theca, first theca, second theca. This lobe also contain 2 theca, first theca and second theca. So overall anther contain 4 theca, so called tetrathecus. And each theca contain microsporangia, 1 microsporangia. This theca contain 1 microsporangia, second microsporangia, third microsporangia and 4 microsporangia. So there are overall 4 microsporangia present in single anther, but in single lobe 2 theca and 2 microsporangia. Clear? Then we discuss about the structure of microsporangia. That means how many layer present in the structure of microsporangia. So the outermost layer epidermis, then inner to epidermis endothecium, then inner to endothecium middle layer, and innermost layer is tapetum. We also discussed that epidermis, endothecium and middle layer help in the protection and dehiscence of anther, and the last layer, which is tapetum, provide nutrition to developing pollen grain. So we discuss about this or discuss up to this. Now we are going to start how pollen grain is going to develop in the sporangia, in this part. As we know, it is theca. Theca contain microsporangia. Microsporangia contains sporogenous tissue, and sporogenous tissue in future going to develop into a pollen grain. So our today's topic is how pollen grain is developed. So let's start with this topic. Wait a minute, I just clean up the board and we'll start with our next topic, which is Microsporogenesis. Don't confuse by hearing this name. As we discussed yesterday, why micro word is used? Micro, that means we are talking about the male gamete, because male gamete is smaller in size, and sporo, that means spore, which is going to develop male gamete. Genesis, that means production. So the process of production of microspore or the process of production of male gamete is called microsporogenesis. Now let's start with the sporogenous tissue. So as we know that innermost layer tapetum, and inside the tapetum the sporogenous tissues are present. So suppose these are sporogenous tissue which is present in sporangia, in microsporangia. Sporogenous tissue. Each and every cell of sporogenous tissue differentiate into the pollen mother cell. Now there are 2 words, 2 similar words. First, differentiation, and second, division. We know the meaning of division. Division, that means when one parent cell simply divide and produced 2 daughter cell is called division. But right now in this topic we are talking about differentiation. Differentiation is very much different from division. Division, that means production of daughter cell, and differentiation, that means to get some specific work. That means cell gets specific work. Now cell know what it want to do or what it need to do. Cell को समझ में आएगा उसको क्या करना है. Done. So differentiation, that means cell gets some specific function. So the after differentiation, of sporogenous tissue, the pollen mother cell is developed. PMC, short form. Full form is pollen mother cell, which look like this. Done, with nucleus. Remember, each and every sporogenous tissues or each and every cell of sporogenous tissue is going to differentiate into pollen mother cell, but we are taking only one pollen mother cell just for better understanding. Clear? So let's start. Now one more important thing, what is ploidy? The ploidy of whole plant is diploid. Diploid, that means each and every cell of plant contain pair of chromosome, not a single chromosome. Pair of chromosome. That means if I say human contain 23 pairs of chromosome, that means human contain total 46 chromosome because there are 23 pairs. Similarly, if I say human contain only 23 chromosome, that means there are only 23 chromosome because there is no pair, each and every chromosome present singly. That's why we called only 23 chromosome, and here we call 23 pairs of chromosome. So when we are using word 23 pair, that means it indicates 2n condition, diploid condition, and when we are using only 23 chromosome, it indicating haploid condition. Simple. In this case, in case of plant, the each and every part of plants, each and every cell of plants is generally diploid, except the gametic cell or cell which is going to develop gamete. So here, whole anther is diploid, the theca is diploid, microsporangia is also diploid, sporogenous tissue or the cell of sporogenous tissue is also diploid, and due to differentiation of sporogenous tissue, pollen mother cell is developed which is also diploid. That means all this structure contain pair chromosome, not single chromosome. Clear? Now next step. In next step, this pollen mother cell undergo meiosis. Most important, undergo meiosis. Now there are two type of cell division. First one is mitosis, second one is meiosis. Clear? Two type of cell division. There are two type of cell division, mitosis and second one is meiosis. Let's discuss it in shallow method so we can understand this topic very clearly. Clear? First one is mitosis and second one is meiosis. What is the difference between mitosis and meiosis? At the end of mitotic cell division, the parent cell and daughter cell contain same number of chromosome. That means if parent cell contain 46 chromosome, then daughter cell also contain 46 chromosome. In case of meiosis, if parent cell contain 46 chromosome, then daughter cell contain 23 chromosome. That means number of chromosome reduced by half compared to parent cell in case of meiosis. This one is first difference. Then in case of mitosis, number of chromosome remains same in daughter cell as well as in parent cell. Second difference, at the end of mitosis, only 2 daughter cell is produced. But at the end of meiosis, 4 daughter cell is going to produced. Daughter cell into 4. So these two are the main difference between mitosis and meiosis which we need to know before understanding this topic. Done. So here which type of cell division take place? It is meiosis. That means if number of chromosome in pollen mother cell is 2n, That means after meiosis, it produces 4 daughter cells, and each daughter cell contain half of the chromosome number compared to parent cell. So as a result, we get 4 cells. After meiosis, we get 4 cells. Each cell contain half number of chromosome. Why? Because the type of division is meiosis. Done. So the ploidy of this structure is N, not 2N. Parent cell is 2N and daughter cell is N. Done. So now we are just roughing this portion, and this structure is called microspore tetrad. Tetrad, that means 4. It's a group of 4 cells. So that's why microspore tetrad. Name of this cell is microspore, but the cells are in the group of 4. So microspore tetrad. Each and every microspore are haploid. Why? Because meiosis take place. Next, after enough time, the each and every microspore from this tetrad is separate from each other. Now there are 4 separate microspore present, like this. And each and every microspore is haploid. All cells are called microspore. And each and every microspore is haploid. Now, this each and every microspore is going to mature into a pollen grain. But we are stopping here to understand what is microsporogenesis in textbook language. So as we can see, sporogenous tissue differentiate into pollen mother cell. Pollen mother cell undergo meiosis to produce microspore. So now we have definition of microsporogenesis. The method from which pollen mother cell undergo meiosis to produce microspore is called microsporogenesis. According to textbook, the process of microsporogenesis include up to this step. These steps combinedly called microsporogenesis. Once again, pollen mother cell undergo meiosis to produce microspore is called microsporogenesis. Now the next topic in textbook is pollen grain, but I am going to connect two topic, microsporogenesis and pollen grain, because वो साथ में पढ़ेंगे ज़्यादा better लगेगा. Now moving towards the next topic, which is pollen grain. Starting from here, each and every microspore is going to develop into a pollen grain, mature into pollen grain, but again we are taking one for better understanding. Done. So we are taking only one microspore, which is haploid. So during maturation into a pollen grain, some kind of change take place in this structure. The first change is it developed two types of outer layer, exine and intine. Let's see. Microspore. Done. The first layer, intine, and the second layer is exine. So this outermost layer is called exine. Inner to exine is intine. This one is plasma membrane for better understanding. Clear? Plasma membrane which is present in all the cell, and nucleus. Clear? This one is also nucleus. And the ploidy of this structure is also haploid. Clear? Next. In next step, the nucleus present inside this cell is undergo mitosis division. Again, mitosis, that means equational division. Number of ploidy remains same, that means number of chromosome remains same, and two daughter cell is produced. But here asymmetrical mitosis will take place. Asymmetrical mitosis, that means at the end of mitosis two daughter cells are produced, but both the cells are unequal in size. If it is equal in size, then it is symmetrical mitosis. But asymmetrical mitosis, that means both the cells are unequal in size. It's look like this. We can write here mitosis. Clear? but type of mitosis is asymmetrical. Asymmetrical mitosis. So as a result of asymmetrical mitosis, two cells are produced, but both the cells are unequal in size. This one is outer layer, clear? Exine, intine, plasma membrane, and inner side. Two types of cell is produced. One is large and irregular in shape, which is called vegetative cell. Vegetative cell. And second one is smaller in size and spindle in shape, called generative cell. Generative cell, clear? So the pollen grain, still it is unmature pollen grain. We are going to discuss how it is going to mature, but type of pollen grain which we are discussing, it contain exine, intine, plasma membrane, vegetative cell and generative cell. And one more structure is there. If you observe this diagram and this diagram carefully, you can observe that at some place exine is absent. See. So the site at which exine is absent is called germ pore, and this germ pore in future is going to help in the development of pollen tube. We are not discussing it right now because it's a quietly different topic, but in shortly germ pore is going to help in the pollen tube development. This one is germ pore. Germ pore. Now if you observe carefully, this pollen grain contains two cells, so it is called bicelled structure. Bicelled structure, and it is haploid. So pollen grain is haploid structure and bicelled structure. But this bicelled pollen grain, it's still unmature pollen grain. How it is going to mature? We are discussing it now. So let's understand how this unmature pollen grain is going to mature. So we discuss about this. Now we are moving forward. Once pollen grain appeared in bicell stage, it has two possibilities. It has two possibilities. One possibility take place in more than 60% of angiosperm, and second possibility take place in about 40% of angiosperm. About 40% of angiosperm. Now what happened? Let's understand. In more than 60% of angiosperm, pollination take place at this stage. That means bicelled stage, at unmature stage of pollen grain. Simple. What happened? With the help of wind, with the help of insects, with the help of water or some other carrier, this pollen grain is spread from the anther and it is going to land on the suitable surface of stigma. And once it's land on the surface of stigma, it is going to germinate and start the process of fertilization. Why? Because androecium is a male reproductive part, it contains pollen grain. Similarly, gynoecium is a female reproductive part, it contains female gamete. So pollen grain needs to reach or needs to land on the stigma to reach up to the female gamete. So simply with the help of wind, with the help of water or insects or through any other animal, it is going to land on the stigma at bicell stage, at this stage. See. This one is pollen grain. We are ignoring the germ pore right now for your better understanding. It is going to land on the stigma at bicell stage. At bicell stage. Once It land on stigma, then it start developing pollen tube. That means germination of pollen grain. When pollen grain is going to germinate, it produced pollen tube. It's looking like this. See. This extended structure is called pollen tube. Now you can easily understand why germ pore is there. Germ pore help in the development of pollen tube. Clear? It is also two cell stage. This one is stigma. Clear? What happened now? This generative nucleus, smaller spindle shaped nucleus, undergo mitosis and produce two new cell called male gamete. See what happened. What happened here? Vegetative nucleus remain as it is, but this generative nucleus undergo mitosis and it produce two new cell, and these both new cell now known as male gamete. And when pollen grain appeared in this stage, that means 3 cell stage, 1, 2, 3, then it is called a mature pollen grain. At 2 cell stage, it is unmature pollen grain. But in more than 60% of angiosperm, pollination take place at 2 cell stage and it is mature after landing on stigma. Clear? So label करते हैं. This one is called male gamete. How it is developed? It is developed through mitosis of generative cell or generative nucleus. And this pollen grain, 3 cell stage pollen grain, 3 cell stage pollen grain called mature pollen grain. 3 cell stage pollen grain called mature pollen grain. What happened in case of 40% of angiosperm? In case of 40% of angiosperm, this generative nucleus or generative cell divide through mitosis before pollination. That means this pollen grain right now it is in 2 cell stage like this. Clear? Now generative nucleus undergo mitosis before pollination, so it become like this before pollination. Vegetative cell and generative cell undergo mitosis to produce 2 male gamete. How it is developed? It is developed by mitosis of generative cell, and this one is also 3 cell stage mature pollen grain, but it is matured before pollination. So we can write this pollen grain mature before pollination in case of 40% angiosperm, but in case of more than 60% angiosperm, pollen grain is mature after pollination. So we can write here also pollen grain mature after pollination. Clear? So here the basic difference, what happened in case of more than 60% angiosperm and 40% angiosperm. Now, let's discuss the functionality and structure of each part of pollen grain. First, vegetative nucleus. It is generally larger in size. Now we know that generative cell is going to undergo mitosis at the time of pollination or before pollination or after pollination, and generative cell is going to develop male gamete. These two are male gamete, these two are male gamete. So male gamete required lots of nutrition. Why? Because once this pollen grain spread from the anther or released from the anther, so he lost his all contact from the anther. So it cannot take nutrition from the anther. So this generative cell needs to take nutrition from someone else. So it is going to vegetative cell. So large in size and provide nutrition to generative cell. Clear? Generative cell is a main cell which help in the development of male gamete. Clear? Exine is the outermost layer. You can see with the yellow chalk. Clear? And it is made up with the help of sporopollenin. Sporopollenin is a very rigid material. It cannot be degraded by majority of the enzyme. So it is very strong substance. Clear? Similarly, intine present inner to exine. It is made up with the help of pectin and cellulose. Similarly, jampur is going to help in the development of pollen tube. Done. So according to textbook, pollen grain topic is just finished up to this. Thank you.